Hello friends, in this video lecture we will discuss the Lac Operon. So friends, we know a particular gene express themselves into a polypeptide by the different basic processes like transcription, translation, etc. etc. And these gene expressions are regulated inside the cell by the metabolic status of the cell, the physiological status of the cell, as well as the environmental status of the cell. But these genes express themselves in a very regulated manner. Whenever they are required, they are supposed to switch on, and when they are not required, then they are supposed to switch off. So this regulation of the gene expression can occur at various level inside the cell, right? Like it can be regulated at the transcription level, it can be regulated at the translation level. Many a times in eukaryotes, they are regulated by the transport of mRNA from the nucleus to the cytoplasm, or many a times it is regulated at the processing level also, right? So here in this video lecture, we restrict ourselves to the prokaryotic system only. So in the prokaryotes, mostly the gene regulation is regulated at the transcription initiation site. And friends, we know the RNA polymerase, it binds at the promoter site, and this binding or interaction of RNA polymerase to the promoter site is also regulated by many more accessory proteins like activator protein or enhancers, sometimes repressors which bind at the operator site. Okay, so the regulatory protein can act both positively like activators as well as negatively like repressors. Okay, so first of all we try to understand, so what is the positive regulation and the negative regulation. So first, the negative regulation. So negative regulation is nothing but if the expression of gene is not there, that means switched off in the presence of a particular protein. See here, this pink protein is responsible for the switching of the gene. No such expression here. So this protein can be considered as negative regulator. Here in another example, this protein itself, when bind to the operator site or the regulatory site, we can say, then there is no expression. But as soon as these proteins leave the operator site here and here, then transcription started, right? So as long as these proteins are there at the site, the gene is switched off. So these proteins are negative regulators, and this process is called negative regulation overall. But the positive regulation is that when a particular protein, like here this green protein is bound at the operator site, and this protein facilitates the binding of the RNA polymerase to bind at the promoter, so the transcription is efficiently occurring here, right? So this protein is considered as positive regulator. But as soon as this protein leaves the operator site, then no transcription at all, right? Here again, this protein as long as bind to the operator site, transcription occurs, right? So this is positive regulator. In nutshell, if a particular protein when bound to the regulatory site and it is responsible for the expression of gene, or it facilitates the expression of the gene by the different means, then it is a regulatory, then this protein is called positively regulatory protein, and this process is called positive regulation, right? Okay, now we try to understand how the lactose metabolism occurs inside the E. coli. So the lactose metabolism in E. coli was observed very fantastically by two scientists, Jacob and Monod, and they published their findings in 1961 approximately, and they first elucidated the transcriptional regulated system, right? So the gene regulation can also occur at the transcriptional level, the first time elucidated. Here, the Lac Operon, Lac refers to the lactose, and Operon, this word comes from the opera. Opera is a kind of play where, the dancer, different musicians, they are working on the direction of a single director. So in the same way, here Jacob and Monod proposed a hypothesis in which they try to explain the polysystronic model of the gene, in which many structural gene has been regulated by a single set of promoter or common promoter, right? Like a common director in the opera, right? So this hypothesis is called the operon, right? After the discovery of the Lac Operon, many such operon like trip operon, arabinose operon, histidine operon has been discovered. So first of all, we try to understand the structure of the Lac Operon. So as the Jacob and Monod said, the set of structural genes has been regulated by a single regulatory element. So here they found in the Lac Operon, three structural genes are there, LAC-Z, LAC-Y and LAC-A, and these three structural genes has been regulated by a single set of regulatory element, a single operator is there, and a single promoter is there, right? So promoter is the site where the RNA polymerase binds, right? And the operator is the site where the regulatory proteins, we call them lac repressor here, is bind, right? Okay, all right. If the expression takes place, then LAC-Z, it produces an enzyme which is called beta galactosidase. The LAC-Y, it produces an enzyme which is called galactoside permease or simply permease, and LAC-A, it produces an enzyme which is called thiogalactoside transacetylase or simply transacetylase. Okay, all right. The function of the beta galactosidase is that it converts the lactose into glucose and galactose, right? Permease, it facilitates the transfer of or entry of lactose from the outside to the inside of the bacteria. And transacetylase, the function is not very well known, but some scientists reported that the toxic effects or toxins produced in the catabolism is neutralized by the transacetylase. Okay. Now, this Lac Operon, right, is regulated by a regulatory protein which is called repressor, and this repressor is synthesized by a separate set of operon which is called LAC-I, having own promoter which is called P-LAC-I. It produces a monomeric repressor, right, after transcription and translation, and this monomeric repressor tetramerize themselves, right? So they are having protein-protein interaction domain. So by that they tetramerize themselves, and after tetramerization they evolve with a binding domain which will interact at the operator site, right? So they develop a domain which is the DNA binding domain, right? And once the repressor bind at the operator, then it would be very difficult for the RNA polymerase to move from the promoter through the operator and come to the LAC-Z, Y and A genes. This figure explains how the lactose enters inside the cell. So as we discussed, the galactoside permease or simply permease, it facilitates the entry of glucose inside the cell. Okay. And inside the cell, the beta galactosidase, it cleaves the lactose into the glucose and galactose. Apart from that, the beta galactosidase have an additional function which converts some amount of lactose into its isoform, which is called allolactose, right? And this allolactose is actually the inducer of the Lac Operon, right? So these are the things which we already talked in the previous slides. So now the we now we discuss how the operon is switch off and switch on. So as we discussed, this tetrameric like repressor have evolved with a DNA binding site, and it can bind at the operator site, right? So if the lactose is not there inside the medium, right? So it can very easily bind to the operator site, right? And once it binds at the operator site, it blocks the movement of RNA polymerase, right? So RNA polymerase is not able to come at the Z, Y, and A gene, right? So no transcription at all of any of the three gene. So no enzyme is there finally. So we can consider the Lac Operon is switch off, right? Now another condition, when the lactose is present inside the cell, in bacterial cell. So as we discussed, the lactose is converted into the allolactose by beta galactosidase enzyme, and these allolactose act as inducer. What happens actually? These allolactose will bind at the tetrameric repressor, and they change the DNA binding motif or domain which is there on the tetramer. So now the tetramer is not able to bind at the operator site. So if they are not binding at the operator site, then RNA polymerase, which already bound at the promoter site, can easily move from the operator and are able to transcribe the Z, Y, and A, right? And finally, the enzymes beta galactosidase, permease, and transacetylase has been synthesized inside the cell, right? So the beta galactosidase, now it breaks the lactose into glucose and galactose. So now we can say the operon or the Lac operon is switched on. So this switch on and off, it totally depends on the presence or the absence of the lactose here, right? Okay. So here the allolactose, which is nothing but the isoform of the lactose, it makes the Lac operon switch on, right? So this operon is called inducible operon, right? Means the substrate itself, lactose is the substrate, and substrate itself makes the operon switch on. So this operon is called inducible operon. Keep in the mind. Okay. Now the question is that when we grow bacteria in a lactose containing medium, right? So lactose is outside the bacteria. So how it can induce the Lac operon, right? Because no permease outside or inside the bacteria, right? So how the first molecule of lactose enters into the bacteria and it converts into the allolactose, right? So the answer is that the basal level of transcription and translation or basal level of protein homeostasis is always maintained inside the cell. So here also the basal level of transcription always occurs inside the cell by a process which is called leaky expression, right? Leaky expression, it means the moment comes inside the cell when the tetrameric repressor leaves the operator, right? Because nothing or no binding inside the cell is permanent. They always keep on binding and leaving the place. So as soon as the tetrameric repressor leaves the operator, so RNA polymerase it crosses the operator and reaches to the Z, Y, and A and completes the basic level of translation. So basic level of all the three proteins are always maintained inside the cell, right? So now if you understood the things, you try to solve this question. This is the CSIR net question. Question is, E. coli mutants isolated from genetic screen showed following classes of mutations. Number one, the point mutation in LAC-I, right? Then the B is the deletion immediately downstream to the transcription start site of the LAC-ZYA. That means they are talking about the operator, right? So point mutation at LAC-I is there, so repressor is not formed, and deletion of operator. That means the repressor has not been bound at the operator site. Then duplication of the part of whole Y and duplication of the whole of, like, it doesn't mean anything actually. It won't affect the Lac Operon. Choose the option which is likely to result in constitutive expression, right? So of course A and B, right? So point mutation at LAC-I, that means it won't produce the Lac repressor, so the operon would be constitutive. Second, if the mutation is there or deletion of operator is there, so again the Lac operon is constitutive because operator cannot facilitate the binding of the repressor. So the choice number A is the correct answer. I mean 1, both A and B is correct. Try to solve this question also. So just I'm telling you the key points of this question. So two E. coli strain is there, A and B, right? And A was earlier grown in presence of the IPTG, right? IPTG is the gratuitous inducer. Then both culture A and B are inoculated in a suboptimal concentration of the inducer, and it was observed that the culture B was unable to utilize lactose, right? Whereas culture A did so efficiently. The reason behind this is, right? So the reason is very simple. Reason is that, the pre-inoculation of the IPTG of culture A, it induces the accumulation of the permease and other enzymes also, but here permease we can say, right, inside the cell to high level. That's why they easily grow in the second inoculation or suboptimal inoculation of the gratuitous inducer. So the if you go through the choice, so third choice, the culture A, the lactose permease was induced to a high level during the pre-treatment, right, which allows the preferential uptake of lactose by the culture A, right? So this is the correct answer. Now we talk about this LAGZ is the basis of the blue-white selection, right? So in this selection, what happens actually, the scientists, they discover a compound, synthetic compound, which is X-gal, and this beta-galactosidase is an enzyme which cleaves the lactose, right? But apart from lactose, it can also cleaves the X-gal into a blue color precipitate, right? And in those bacteria where the LAGZ gene is there intact, they can change colorless X-gal into the colored blue precipitate. So this portion we discuss in more detail in RDT chapter. Now we talk about the Lac promoter, and then in the next slide we discuss about the Lac operator. So Lac promoter is a weak promoter, right? Because if we look at the consensus sequence of the promoter sequence, so this is TATAAT, right? But here in the Lac promoter, the sequence is TATGTT. So this promoter is not exactly similar to the consensus sequence of promoter, right? So this promoter is considered as weak promoter. So RNA polymerase binds weakly to the promoter region, but There is another site which is called CRP site, right? And at this site, enhancer molecule, which is nothing but a complex of CMP CRP, right? From where this complex comes, we discuss in the later part of this discussion. So this CMP CRP, when binds to the CRP site, that means enhancer site, so now this promoter becomes strong promoter, right? So the Lac promoter is a weak promoter. It binds RNA polymerase weakly until the polymerase is activated by the CRP CMP complex, right? Now the Lac operator. So three different operators actually present in the lactose operon. The main operator is called O1, right? Apart from that, two subsidiary operator O2 and O3 is also present. O2 is present in the downstream of the O1, and it is exactly inside the structural gene of the lactose operon, whereas the O3 is present in the upstream. If we look at the sequences of these three operators, so the minor changes are there in the O2 and O3 as compared to the O1. Then this lower figure, this figure is nothing but the fold repression of the presence of the three operators is there. So if all the three operators are working well, then the fold of repression is 1300. But if no operator is there, then fold of repression is only 1. And if you look at the the O1 only, so it represses 18 fold, right? So here the message which convey is the O1 is the main promoter and O2 and O3 is the subsidiary, sorry, operator, right? Subsidiary operator, and O1 is the main operator. And these three operator will bind with the tetrameric repressor by forming a loop like structure, right? Now, as we discussed, so Lac Operon is the operon which is regulated negatively as well as positively. So now we discuss how the Lac operon is negatively regulated. So suppose we make a scenario in which no lactose is there, right? So if no lactose is there, so no allolactose at all, right? So the repressor bind to the operator site, of course. Here you can see repressor can bind to the operator site, okay? So when the operator site is blocked, so like operon is repressed, okay, because RNA polymerase is not able to move on to the ZYA. So presence of the repressor, see the presence of the repressor on the operator, it switch off the operon, correct? That's why this is called negative regulation, right? So this Lac repressor, it negatively regulates the Lac operon, right? Okay. Another is a positive regulation. So it has been seen that apart from the lactose, the Lac operon is also affected by the available amount of glucose inside the cell, right? This is very important. And one more thing, you try to understand, remember that glucose is the repressor of the enzyme which is called adenylate cyclase, right, which synthesizes CAMP. Okay, glucose is the inhibitor of the adenylate cyclase. You remember this. Okay, now, so here another situation is there. If the glucose is absent, okay, so in the absence of glucose, adenylate cyclase is activated. Okay, so CAMP has been synthesized inside the cell, and CRP is already there inside the cell. So the CRP, when mix the complex with the CAMP, now it can efficiently bind at this site, right? I mean CRP site, and this is a positive regulator, and it facilitates, I mean CMP CRP complex at the CRP site, it facilitates the binding of RNA polymerase at the promoter, because now the promoter is no longer weak promoter, and it synthesizes all the three enzymes led by A of the Lac Operon, right? So due to the presence of the CMP CRP complex, the operon is switched on, right? So this is the positive regulation, right? So the CMP here is called coactivator, whereas CRP is called apo-inducer, correct? Here, both lactose and CMP CRP complex, they affect, right? Lactose, it negatively regulates, and CMP CRP complex positively regulates, right? Both work independently, but for proper execution of the lactose operon, both will act in concert, right? Both will act together. So now if you understood these things, now you can able to solve this question. Just to read the question, I'm telling you the key points of this question, that Lac Operon E. coli is controlled by the Lac repressor, of course, right, as well as the CAP, catabolite activator protein. So in vivo experiment has been done, and following observations has been made. The first observation is that CMP level are high. What does it mean? If CMP level are high, that means no glucose is there, right? The second observation, the repressor is bind to the allolactose, means lactose is there. That's why allolactose is there, and lactose bind to the repressor. Yes. Third observation is cap is interacting with the RNA polymerase. Okay, that means cap, it will make a complex with CMP, so that it interacts with the RNA polymerase and make it, or bind with the promoter very strongly. So which of the following conclusion is most appropriate based on the above observation? So of course glucose and lactose both are present, right? So glucose, sorry, not this choice. So I'm sorry, so glucose is absent here because CMP level is high. So glucose is absent and lactose is present. So fourth number choice is the correct answer. Okay. Now we discuss the different scenarios depending upon the carbohydrate sources, right? So we here take four different conditions. The first condition is if the glucose as well as the lactose, so glucose is present in the medium and the lactose is absent in the medium. If this is the scenario, so if glucose is present, that means it inhibits the adenylate cyclase, so no CAMP is there, so the operon is switched off, right? And this is an example of negative regulation, right? Because the CMP CRP complex has not been formed, right? And if CMP CRP complex is not formed, then it is weakly expressed, right? So in case of the glucose, which is the favorite carbon source for the bacteria, right? So this is, or this operon is switched off. The second condition, where the glucose is absent and the lactose is present. So if glucose is absent, of course the the adenylate cyclase is activated, so CMP is present, CMP CRP complex is formed, and it will bind to the CRP site. So this is positive control, and it enhances or make the efficient binding of the RNA polymerase, right? This is one thing. Another thing is that lactose is present, so it converts into allolactose, then it binds to Lac repressor and it makes the operon switch on, right? So this is no doubt the example of inducible operon. So the operon is switched on, and due to the presence of CMP CRP complex, it is positively regulated. So this is second situation. The third case is that this is a very interesting case. So glucose is present and lactose is also present. Both are present. So see, if glucose is present, that means no CMP CRP complex is formed, right? Now you can understand. So no RNA polymerase binds efficiently, right? Or RNA polymerase can bind weakly to the promoter. Okay. Lactose is also present. So it change into the yellow lactose, then it binds to the lac repressor and it makes the switch on. Okay. So see, both are the contradictory situation here. So if the glucose is there, so first they utilize the glucose. Bacteria will utilize the glucose, right? Because the operon is very weakly expressed. Okay, almost no expression is there when the glucose is present. It is almost switched off, right? But as soon as the glucose is over or finished into the medium, then again the culture acclimatize themselves to use the lactose. So a very small lag phase has been observed in between the two log phase, right? And then again it utilizes the lactose. That means the operon is now switched on. So this kind of graph we get, right? And this is called Diauxic growth curve, right? So when the two carbon sources, a very simple carbon source or favorite carbon source, glucose as well as lactose is present, then we get this kind of graph. Or apart from lactose, other carbon source also maybe, right? Fourth case is very interesting, in which glucose is absent as well as lactose is absent. So logically, this operon should be switched off, right? This is correct also. But when we go through the mechanism, so what mechanism tells us, if no lactose is there, so no yellow lactose, so repressor bind to the operator and switch off, right? This condition fulfilled. But the another thing, if glucose is nowhere or absent, then high CMP is there, as we discussed earlier, right? So the CMP CRP complex has been formed. Hence, it makes the operon switch on, or it will try to make the RNA polymerase bind efficiently at the promoter site. But it is not possible. Why? Because the repressor or the tetrameric repressor, like suppose if we make a this kind of diagram, so this is O1, right? Here O1 and here O2 and O3 is there, right? So it will bind the tetrameric repressor first of all, right? And if we talk about the CRP site, right? So CRP site is upstream. Here the CRP site is there, right? And if we talk about the promoter, right? So here promoter is there. So what happens actually? The promote RNA polymerase binds, of course, but it won't be able to move because at the operator site, the tetrameric repressors are already present and it makes a loop-like structure, right? So RNA polymerase is not able to move onto the operator or through the operator to the ZYA. So the entire operon is switched off, right? So if you understood this thing, then you can solve this question now. Just I'm highlighting the key points. So E. coli grown in 3 different medium, right? The glucose as a carbon source, second medium containing glucose and galactose, and third is the infected phage, right? Phage viruses, right? So this kind of graph, right? ABC graph they found, and upon this observation, the choice has been given. So see, just we have discussed the A graph. A graph is looks like Diauxic, right? So certainly two different sugars would be there. One is glucose and another is galactose. This one, right? B is glucose because simply it utilizes, and when glucose is over, then the growth curve is flattened. Okay, and so B is the glucose, right? So A is the glucose and galactose, B is the glucose only, and C is the phage because after some time it will lyse, bacteria will lyse, and finally the population will be, number of cells will be less or zero, right? So the second number choice would be the correct answer, right? Now we discuss the lac mutation analysis. This is very important. So now the Jacob and Monod, they try to understand whether and how or which are the different situations in which the Lac Operon is constitutive or uninducible, whether it is dominant or recessive, cis acting elements are acting or trans elements are acting, right? Whether the the operator or promoter or regulatory elements or structural genes are recessive or dominant, etc., etc. So for that they made merozygote, and merozygotes are nothing but the partially diploid bacterial cell, right? So what they did actually, they developed a merozygote of the Lac Operon. So this is the chromosomal DNA and this is the plasmid DNA on which the Lac Operon is there. And they studied various mutants of the merozygote. Here we discuss four different conditions of the merozygote. So first case is that in the Lac Operon, the E. coli with mutation at the regulatory gene. So this mutation is I minus mutation. So this is merozygote condition. This is the wild type gene and this is the gene present on the plasmid. So plus it indicates the wild type and minus here is the mutant. So I minus means it won't synthesize any repressor, or they won't synthesize repressor protein. So the repressor protein has not been synthesized by the merozygote, but the wild type gene, this synthesizes repressor, the tetramerase also, and they bind at this promoter as well as this promoter with the same efficiency without any difference, right? So here in this operon also, the expression of the lac gene products are not there, right? Here not there and here also not there, right? So what we can conclude? So here we can conclude this I minus mutation is the recessive mutation, right? Because this I minus mutation is not able to even stop the expression of, or the execution of the, or it won't be able to make the switch on this operon also, right? So this is the recessive mutation because it is fulfilled by this I plus. Okay. Now the second situation is that when a mutation has been done at the operator site, right? So this is the OC operator, mutation has been done. So what happens here? Both I plus and I plus, they synthesizes tetrameric repressor, but these tetrameric repressor can bind here, right, on the wild type DNA, but it won't be able to bind at the merozygote DNA, right? So merozygote DNA, the structural gene of the Lac Operon, it expresses, right? Whereas the repressor binds at the original DNA strand of the Lac Operon. So here the expression is not there, right? So what conclusion we can make from this observation? So we can make here the Lac Operon here, or we can see the operator is cis acting, right? So this operator is acting only on the merozygote DNA strand. So this operator can be called as cis acting element, and this mutation makes the merozygote DNA switch on, right? So this is called dominant kind of mutation because this cannot be interfered by any other gene or gene products of the original strand of the DNA, right? This DNA, isn't it? So this mutation is called cis dominant mutation. Okay, dominant, why? Because the gene expression of this DNA or the Lac Operon expression of this DNA cannot be halted by the upper strand of the DNA. Okay. Third condition is that the mutation has been made in the regulatory gene, again the I gene, but here the mutation is different from the first case. Mutation has been done in a way that the inducer, I mean the allolactose or IPTG, cannot bind at the tetramer, right? Mutation has been done in that way. So what happens? See here, IS, right? So IS, it synthesizes a repressor which is yellow in color here, right? And the repressor synthesized by the I plus is green in color, right? So both repressors are synthesized in the same amount, right? So here the tetramer has been formed by the combination of both green and yellow repressor, right? And the mutation in the green repressor is that inducer cannot bind, right? So it overall, I mean this overall cannot be affected by the inducer when entire tetramer, whether only one or two or three yellow repressor is there, it is not affected. It always binds at the operator site. Whether the inducer is there or not, it won't affect, right? So presence or absence of the inducer, in both case, it will bind at the operator site in the wild type as well as the Merozygote DNA, right? So the gene expression is off in both the cases. So now the observation, which kind of conclusion we can draw? So see, this mutation is, first thing is the cis as well as the trans-acting mutation, because the mutation has been done on this strand, but the gene expression has been switched off this strand, I mean this DNA as well as this DNA. So this is both cis as well as the trans acting element or mutation, right? And this mutation cannot be fulfilled by the any other gene which is present in this combination. So this is again called dominant. So this is cis as well as the trans acting dominant mutation. Okay, try to understand. Okay, think about that. Okay, now we discuss the fourth case. Fourth case is that again the mutation has been done on the I gene, but the mutation is different here. This mutation is I minus D, right? And this is called dominant negative mutation. So in this mutation, what happens? It forms a tetramer, right? Again, here the yellow color repressor has been synthesized by the minus ID, right? But the thing is that after formation of the tetramer, they change the DNA binding domain of the tetramer. So they are now not able to bind at the operator site, right? They cannot bind at the operator site. So what happens? Both the gene, right? This wild type as well as the merozygote will express themselves, right? So which conclusion we can draw here? So this mutation can be called dominant mutation, of course, because this mutation cannot be supplemented or fulfilled or corrected by the any gene of this merozygote. So this is of course dominant, right? And the another thing is that this is cis as well as trans both, right? Cis acting as well as trans acting. And another thing is that what happens actually, why it is negative. So what happens here, the mutant protein, it poisons the activity of the wild protein, right? So this protein, this wild protein, green one, right? It also becomes poisons due to the addition of one or two or three molecules of the green repressor protein. That's why this is called dominant negative, right? This is nothing but the summary of the different cases what we discussed earlier, right? So just you go through the slide and try to understand here in the presence of beta galactosidase as well as in the absence of the beta galactosidase, cases has been given and all the cases are written here. The conclusion has been written here, right? If you understood these things, then you can be able to solve this question, right? So what the question is, just I'm reading the key points. So a merozygote has been synthesized in which OC, ZY, Z minus and Y plus, whereas the O plus, Z plus, Y plus is there, right? So this is the merozygote. And the activity of beta galactosidase enzyme was measured upon the following treatment, right? Okay. So the first stage, no induction, when no allolactose or lactose is there. So what happens in this case? And we always think about the beta galactosidase enzyme activity, right? Because the answer is given in the form of graph, right? So see, no induction means the OC, the first case, see here, the OC, I mean, the operator is mutant, but here what happens? Z is minus. That means they won't be able to synthesize the beta galactosidase. So no beta galactosidase is there. And second case, here O plus is there, right? O plus means wild type operator is there, right? And wild type operator, it always occupies with the tetrameric repressor when the inducer is nowhere or not there, right? So if induction is not there, basal level of transcription and translation takes place. So some amount of beta galactosidase or very less amount of beta galactosidase would be there, right? So first bar should be the less, not zero, because zero is not there because always basal level of transcription is there. So this and this may be the case. Okay, now we move ahead. The induction with the n mole of IPTG. Okay, so IPTG is the inducer. So of course, the level of the beta galactosidase would be higher than that of the first case, right? So again, both cases, the beta galactosidase, the activity is higher than that of the A case. Okay. So third, the induction with the n mole of lactose. So n mole of lactose, that means in this case, see, the lactose is catabolized, right? But IPTG is not catabolized. So the bars should be lesser, height of the bar should be lesser in the case of lactose because it is catabolized after some time, but IPTG is not catabolized, right? So the option 3 would be the correct answer here. But we should, read the fourth option also, fourth condition also. So induction with the n mole of lactose in the presence of the n mole of glucose, right? So see here, the lactose and glucose both are there. So what happens here? The expression would be there, right? N mole of lactose as well as the n mole of glucose is there. So certainly the expression would be there, but less amount of expression would be there because glucose is there. Okay, so glucose is there, so the no CMP CRP complex will formed. So answer 3 is the correct answer. So friends, if you understood this entire lecture, so please like the video, share with your friends and colleagues, and subscribe so that we can boost up to make the other videos also. So thank you very much.